Hello, welcome back. Today's video is going to be on construction of a standard curve and linear regression analysis of the data. One of the cornerstones of experimental analysis is the ability of a researcher to acquire data and uh, convert the acquired data into the amount of a substance that is being analyzed or amount of the analyte that is being analyzed. And in this regard, often when a sample is analyzed for quantitative analysis, parallelly a standard is also constructed. And I hope this video will be useful for those who are trying to learn how to construct a standard curve and analyze the data by linear regression. Please keep in mind that linear regression is not the only way a standard curve data is analyzed. Often, we also have to resort to non-linear regression analysis, particularly the ones who do LI so often have to look at non-linear regression analysis of the data. But in most simple experiments, the output of the experiment often follows uh, a linear correlation with the analyte being analyzed. So that's why linear regression analysis is done. So in this regard, what we'll do is we'll look at a data set and try to construct a standard curve, and then look at a few considerations, and then try and do linear regression analysis, and then use the parameter from the linear regression analysis to be able to determine the amount of substance in an unknown sample, right? So the data set that I have displayed here is actually the absorbance that was obtained for different concentrations of standard protein, that is bovine serum albumin, taken in an experiment which was conducted following the method of Lowry et al. Okay, so the data was acquired, and the data is depicted as such. When the data was acquired, there was no blank correction. I'm sure those who do experimental analysis will know what it means to blank correct the data. I mean, especially if you're looking at a spectrophotometer, then you would often do an auto zero where a blank is taken. Blank is the entire assay reagent except the analyte being analyzed. Uh, it is used to set the spectrophotometer at an absorbance value of 0 or transmittance value of 100% at a given wavelength. So I did not do that primarily because the data was acquired using a microplate reader. So the sample was placed in a microplate plate, and then the data was acquired. So this is the raw data. So we have to transform it by doing blank correction manually. So what we'll do is we'll just so this is the absorbance value for blank. Okay, so we'll just correct this, correct all the absorbances for different concentrations of protein using blank correction. So I'm just going to subtract all the values, including the blank value, with 0.077. Okay, so that will give me a blank corrected value. Okay, so I'm going to just drag the parameter down over all the columns. So now I have blank corrected values. So I'm going to just copy these values and paste it over some other cells, and we'll just juxtapose these values to the concentrations of the protein. Okay, now what we've done is we've just transformed the data by doing blank correction, and then we have the data set ready for regression analysis. Okay, so the first column is the concentration of protein. Second column is the absorbance values corresponding to each concentration of protein. So I'm going to drag over these two columns, the entire data set, and go to insert option and insert a, from the option of charts, a scatter plot. Okay, now if you can see the plot carefully, and this is one consideration I wanted you to appreciate. If you can see this, on the x-axis we have the concentration of protein, and the y-axis is the absorbance values at 750 nanometer. Now if you can see that beyond a certain concentration, uh, the absorbance values do not linearly correlate with increase in the protein concentration, and this is typical for Lambert-Beer's law. We all know that uh, the linear relationship only extends up to a particular concentration, beyond which the linear relationship is lost when you're looking at UV visible. I mean, when you look at absorption of UV visible radiation by chromophores. Okay, so typically what I do is when we have graduate students, we ask them to consider standard curve, uh, taking concentration range which is way well beyond linearity, and we want them to appreciate the limitation of Lambert-Beer's law as far as a certain threshold of concentration is concerned. Okay, so it would be absolutely wrong to take the entire data set for linear regression analysis, simply because beyond certain concentration there is no linear relationship between the absorbance value and the concentration of the protein. It implies that you should always, when you do a sample analysis, your sample value should be within the standard range. If the sample values are higher than the highest value for standard which follows linear regression, then we'll have to dilute the sample. Okay, and this is one experiment or one exercise that would serve very well when done, taking concentrations beyond linear range. It will serve well for students to understand what truly Lambert-Beer's law is that underlines the principle of UV visible spectroscopy. Okay, so we can't do regression analysis over the entire concentration range, so we'll have to limit our study or our analysis to a range of concentration which gives us fairly linear regression. Okay, so what I'll do is I'll still plot a graph. I'll add a trend line. Okay, so in order to in order to do linear regression, first you have to add a trend line. Okay, just add a trend line. You'll get options here to choose from. Because we have done blind correction, I'm going to set the graph to pass through x and y is equal to 0. I'm going to set the intercept x, y is equal to 0, and I want to see the equation on the chart, and also I want to see r squared values. So r squared values is a measure of how close the points are to that regression analysis line. Okay, now as you can see, the data set is pretty bad. Okay, it's horrendous. Now what I wanted to appreciate is I'm going to just place the cursor over the graph, which will allow me to select the data set, and I'm going to just decrease the range of the concentration to see up to what point we get satisfactory visual observation in terms of satisfactory linear relationship between the two variables, right? The two parameters. So I'm going to just reduce the range. Let's see. Okay, still not very linear. Okay, still not very linear. Fairly okay, but I wouldn't accept it. Although R square value is good, I still will not take it. I'm going to decrease it further. So I'm going to only do an analysis up to 0.8 mg per ml. So from 0.1 to 0.8 mg per ml, there is a fairly good uh, regression, uh, linear regression, fairly good linear correlation between the absorbance values and the concentration of the protein. And what I'd like you to look at are two things. One is why is this why is the equation for the line. Okay, typically a straight line will have the equation y is equal to mx plus c, because the line is passing through intercept. The c value, which is the y intercept, will be 0, so it's just y is equal to mx, and the number that is shown here, 0.816. Okay, you can look at this graph as y is equal to mx, and I mean, if you can recall, or if you can refer to some material reference literature, you will realize that the m value, or the number that is shown here, is the slope of the line. The slope tells you, uh, uh, how the y varies in relation to x. Okay, now that is very important. And the next thing that you got to keep in mind is the r squared value. Okay, for most experimental analysis to be valid, you need to have an r square value of as close as possible to 1, preferably above 0.99. Okay, 0.98 is also accepted, but less than 0.98 is very bad. You shouldn't rely on that data, and you have to repeat the experiment altogether another time, so that your pipetting skills are accurate, and you're doing experiment properly without introducing manual errors. Okay, so you've got to practice until you're able to achieve an R square value of as close as possible to 1 above 0.99. This is fairly okay. I'll just tweak the values on the absorbance column to show you how R square values are going to get worsen as there is deviation from the trend line. Okay, so let's say I'm going to change this value to zero point 0.1. I'll assume that there was an experimental error, and instead of 0.181, you obtained an absorbance value of zero point 0.1. See how the R square value decreases now, seeing that it's now 0.941 because this point has gone far away from the trend line, right? So it tells you that there is an error. Okay, and I'm going to increase, let's say, this value to zero point 0.6. Okay, so now these 2 points represent experimental errors, and the more errors we have, the more the farther the points will be from the trend line, and as far as they go, the R square value gets compromised, and we don't want that to happen. The R square value should be as close as possible to 1 above 0.99. Okay, I would say that you set your own benchmark and do not accept any R square values less than 0.995. Okay, that should be good. Okay, so I'm going to go back to my original data set, and now we have a fairly linear graph in terms of the increase in absorbance as a function of protein concentration is concerned, and R square values are also pretty good, and we also have the slope, right? So now let's see how we can use this graph to determine the amount of protein in an unknown sample. Okay, let's hypothetically assume that the unknown sample that you tested for the protein concentration gave you an absorbance of, let's say, 0.31 or 323. Okay, now remember, this is the raw data that is acquired. So I still have to subtract this with the blank value, which, if I can remember properly, it is 0.077. Okay. So now this is blank corrected value for unknown. Now you just have to divide this unknown by the slope. Okay. In our case, the slope is 0.8164. So if you divide that, you're going to get the protein concentration. Okay, the protein concentration of our sample is 0.301 mg per ml because it had an absorbance of zero point 0.2. Okay, 0.246. It's as simple as that. Okay, so this is how we would determine the uh, 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 concentration of an analyte using standard curve. Hope this video was useful in your understanding of how standard graph is constructed and how regression analysis is done. Linear regression, mind you. Okay, as I told you earlier, there are a lot of experiments where you cannot do linear regression analysis. Okay, you have to go for non-linear regression analysis, particularly if you're doing ELISA and or receptor binding studies. In such cases, you have to go for non-linear regression, but simple experiments will often follow linear. For simple experiments, linear regression analysis is enough, assuming there is a linear correlation between the output of the experiment and the analyte concentration which is being measured. Okay, so this is how you would determine the amount of an unknown protein using, unknown analyte. Okay, using the standard curve by doing linear regression analysis. I hope this video was useful. If it was, then please do subscribe to my channel. It will only encourage me to post more videos. Thank you very much.